All right, today's lecture is going to be on the neural system. We're going to talk about an introduction to the neural system, and I'd like to start with this, silly picture. And the only reason why I put this up there is because if we look at Homer Simpson here, H.J. Simpson, you'll see that the joke is that he has this small brain. So the assumption that a large brain means that it's smarter and faster, and that a small brain is equated with slowness or dumbness is just wrong. In fact, in the human brain, sometimes the smallest part of the brain is the quickest and the fastest. And just think about computers. Think about when computers started, how large they were, and they needed all these cooling AC units and fans to cool them off. And now computers are getting smaller and smaller and smaller, and phones are getting smaller and smaller, and yet they're getting faster and faster. So just food for thought on that. When we look at the organization of the neural system, we'll see that we have the CNS, which is the central neural system. And when you hear CNS, we're thinking brain and spinal cord. When you hear of the PNS, the peripheral neural system, that's everything else. That's the nerves coming off the brain, cranial nerves. That's the nerves coming off of the spine, spinal nerves. Okay? And there is information that comes in to the brain and the spinal cord. We call that sensory information or sensory input. And then we have motor information that comes from the brain down the spinal cord, and then it goes outward. So special senses that come from the eyes and the nose and smell and taste, etc., and senses from the soma, they come in to the brain and the spinal cord, and then we have output messages that will then go to the somatic neural system, going to the muscles, right? Muscles are sensitive, they're sensory, they're highly adaptable. If a muscle gets damaged or overly stretched, we know they can react. So now motor information that's received, right, sensory comes in, motor comes out to the skeletal muscle that controls movement. This is voluntary, this is conscious control right here. But motor output to the ANS, to the autonomic neural system, that's involuntary. And there are two major branches of the ANS, two major ones that you're going to hear a lot of, and one is the sympathetic and two is the parasympathetic. Okay, there is a third branch, the enteric neural system. This is like an inborn inherent system to the smooth muscle and the glands of the GI tract. The sympathetic and parasympathetic, we typically think that the sympathetic is like the accelerator and the parasympathetic is like the brake. And for the most part, that's true. Sympathetics tend to speed everything up except for digestion. And parasympathetics tend to slow everything down except for digestion. It actually speeds it up. And the sympathetic is also known as the thoracolumbar outflow or thoracolumbar output because it stems from T1 to L2, so it's called a thoracolumbar outflow or output. And this is known as flight or fight response. Whereas the parasympathetic, this is called the craniosacral outflow. And that's because it's cranial nerve 3, we write it as Roman numeral, cranial nerve 3, cranial nerve 7, cranial nerve 9, and cranial nerve 10, which is the vagal nerve. So we have cranial nerve 3, 7, 9, and 10, and then sacral segments S2, S3, and S4. S2, 3, and 4 help keep the feces off the floor. So craniosacral is feed and breed. It's the calming system, right? It helps to explain why when you're eating food, which is a parasympathetic thing, you shouldn't be nervous, anxious, driving in a stress response, arguing with someone, right? When you're having an argument, you don't want to have a meal and digest your food. And when you're exercising, right, or let's say you just eat a meal, you don't want to go swimming right afterwards, right? When your parasympathetics are active and you need blood in the GI tract to absorb the nutrients, you don't want to exercise and divert the blood to your skeletal muscles. Okay, so sympathetic and parasympathetic. Most diseases are from an overdriven sympathetic neural system. There are very few that I can think of that are parasympathetic diseases. Okay, let's see if we can get this video running. Oh, it's not a video, it's just here is the brain, here's the spinal cord, these are cranial nerves, and these are spinal nerves. Let's click on the brain, and we'll see that we have some major portions to it. Let's start here at the cerebrum. That's the largest portion, right? There's the cerebrum, not to be confused with the cerebellum. And then we have the brain stem, which is from superior to inferior. We have the midbrain, the pons, and then the medulla. And then we have a diencephalon that's made up of the thalamus and hypothalamus. Again, thalamus, hypothalamus. And then we have these chambers called ventricles, which is where the flow of cerebral spinal fluid travels. Okay, let's go back here. Let's look at the spinal cord. And we're going to get into this in a lot more detail, but here's the spinal cord, and we can see that what protects it are the meninges. And there's 3 layers of meninges. I know you've heard of meningitis, where it's inflammation of these. There's the pia mater, which is the innermost layer closest to the spinal cord. Then there's arachnoid, and then there's the dura mater, which means tough mother. That's closest to the vertebrae. Here are the spinal nerves. And these spinal nerves have a sensory and motor component to it. Subarachnoid space, so here's the arachnoid, and then underneath it, subarachnoid space. That's where the cerebral spinal fluid actually flows. And then there's a dura mater, and underneath that, there's a dural space. Let's look at cranial nerves. There's 12 pairs. And they're numbered by Roman numeral 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, and 12. And they're numbered in order of their embryological development. So the olfactory nerve is 1, optic nerve is 2. Olfactory is for smell, optic is for vision. Oculomotor is involved in moving the eye. Trochlear moves the eye as well. And so does number 6, the abducens. It abducts the eye, it moves it. Trigeminal is sensory to the face, as well as muscles of mastication, which are your chewing muscles. Facial nerve for facial expression, vestibulocochlear for balance and hearing, and glossopharyngeal muscles of swallowing. It also does the taste of bitter to the posterior 1/3 of the tongue, whereas the facial nerve does sweet, sour, and salty to the anterior 2/3 of the tongue. The vagus is about 80 to 90% of parasympathetic control, controls the heart, the lungs, the stomach, the spleen. Here's the accessory nerve. Cranial nerve 11 controls the trapezius and the SCM muscle, and hypoglossal controls the tongue. Again, we'll go through these when we cover the brain, but it's good to at least hear them now and get a jump start on these, because when we do cover the brain, you will have to know the names of these cranial nerves, their Roman numeral, as well as their function, and actually what parts of the brain they come from, and for very good reason. And then we have spinal nerves. We have, here's the medulla, here's 8 pairs of cervical nerves, we have 12 pairs of thoracic, 5 pairs of lumbar, 5 pairs of sacral, and 1 coccygeal. Okay, and so there are actually 31 pairs of spinal nerves. Okay, so we covered everything here. Let's go back.
The nervous system is structurally divided into 2 subsystems. Association neurons, located entirely in the brain or spinal cord, make up the central nervous system. Receptors and sensory neurons and motor neurons in nerves make up the peripheral nervous system. The peripheral nervous system consists of nervous tissue outside of the central nervous system. It is divided into 3 different functional divisions: the somatic, autonomic, and enteric systems. The somatic nervous system contains sensory neurons that convey position information on muscles, as well as motor neurons that convey information to skeletal muscles only. The autonomic nervous system contains sensory neurons that convey information from visceral organs, as well as motor neurons that convey information to cardiac muscle, smooth muscle, glands, and adipose tissues. Most ANS effectors are governed by opposing divisions of the motor portion of the ANS. The enteric nervous system contains sensory neurons that convey information from digestive organs, as well as motor neurons that stimulate smooth muscular movements and secretory activities of the digestive organs.
All right, let's continue on here. Let's look at the neuron, nerves, and neuroglial cells, okay? So here we're looking at the neuron and different parts of the neuron and the nerves. So first we have to look at certain definitions. So when we look at what is a nucleus is a group of nerve cell bodies within the central neural system. A ganglia is a group of nerve cell bodies outside of the CNS or in the PNS. Then we have a nerve and a tract. And a nerve is a group of axons outside of the CNS, whereas a tract is a group of axons in the CNS. So you should kind of know those definitions, like what is a nucleus, what is a ganglia, what is a tract, and what is a nerve. It's like real estate, location, location, location. So if you have a group of nerve cell bodies in the CNS, that's a nucleus, and if you have a group of nerve cell bodies in the PNS, that's a ganglia. If you have a group of axons in the CNS, it's called a tract, and if you have a group of axons in the PNS, it's a nerve. Okay, so when we look at a neuron, which is here, we could see that we have dendrites, cell body, a nucleus, and this little hill, the dilated part of the axon called an axon hillock. Now, the axon is this structure here, this entire structure, and we can see that this axon is covered by myelin. And what does the myelinating is this Schwann cell. This Schwann cell is a neuroglial cell or neuroglial cell that surrounds the nerve fiber and wraps around with this fat, this cholesterol called myelin. And the gaps or the spaces between each of these Schwann cells is called the node of Ranvier, okay, which is actually unmyelinated. So these action potentials go very fast where they're myelinated, and then they jump very fast, and then they jump over the node very fast, and then they jump very fast, and they continually jump. Okay. At the very end of the axon is called the axon terminal, and at the very end of the terminal are those end bulbs, those synaptic end bulbs. Here are the axons, and you can see that the layer that surrounds it is called an endoneurium. Remember, in muscle we had a endomysium. Well, around here is an endoneurium. Then we have a perineurium. In muscle it was a perimysium. And then we have an epineurium surrounding the whole thing. In muscle it was an epimysium. Remember those? Okay. Okay, I think that was it, right? We covered everything else here. Let's go to neuroglial cells or neuroglial cells. Now here we have a few different types. When we look at a microglial cell, here, the microglial cell, it originates from white blood cells, and it protects the CNS. It's involved in phagocytosis. It's like a macrophage. We have these ependymal cells here that's responsible for creating CSF, or cerebral spinal fluid. So we find these all throughout the ventricles of the brain, the first, second, third, and fourth ventricle. We have an astrocyte, astro meaning starlight, and this makes up the blood-brain barrier, and it helps to maintain the appropriate chemical environment around neurons for the generation of action potentials. Then we have a Schwann cell and oligodendrocyte. Now the Schwann cell and oligodendrocyte both have similar function. They produce myelin. The only difference, again, real estate, location, 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 where are they going to do it? The oligodendrocyte here myelinates in the CNS, whereas the Schwann cell myelinates in the peripheral neural system. If there's damage in the PNS, the Schwann cells can remyelinate, but if there's damage in the CNS, in the brain and spinal cord, they cannot regenerate myelin within the CNS. That damage is unfortunately permanent. And these satellite cells right here are they're like supporting cells in the PNS. Okay, let's close this out. So the neural system has three basic functions. Its job is to sense changes within the environment. So we have these sensory receptors, such as feeling full, right? Or knowing that maybe you have a sunburn, or something's hot, or something's cold. That's the responsibility of these different receptors. Then your neural system has to process that and interpret that information and remember it for the future. And when it interprets that information, it then sends that information out and creates some sort of reaction to the effector. And perhaps one of the effects is a muscle contraction. Maybe another is glandular secretions. So again, in summary, the brain and the spinal cord makes up the central neural system, and everything else is the peripheral neural system. So if we're talking about cranial nerves and spinal nerves, that's PNS. If we're talking about the ganglia, which is a group of nerve cell bodies in the PNS, right? We have the enteric plexus in the small intestine. And then we have these receptors, sensory receptors in the skin. They're all part of the PNS. And this is very similar to that first chart that we looked at, where you have the brain and the spinal cord, and we have sensory information coming in either from sensory receptors or muscles or from our organs. And then our brain and our spinal cord will process that and send motor information out to the skeletal muscle, or it'll send motor information out to the smooth muscles, the cardiac muscles, gland, and fat. And those motor signals are either going to be sympathetic or parasympathetic. Sympathetic is fight or flight response, the thoracolumbar outflow, and the parasympathetic is the feed and breed, and it's the craniosacral outflow, cranial nerve 3, 7, 9, and 10, and sacral segment S2, 3, and 4. The CNS is the brain and spinal cord. PNS is everything else. Somatic means it's voluntary. The autonomic means self-regulating or self-law. And again, here's that information. Somatic is motor neurons to the skeletal muscle. The autonomic, now we have the sympathetic and parasympathetic division. Sympathetic is fight or flight, thoracolumbar, T1 to L2. Parasympathetic slows down the heart but speeds up digestion. Sympathetic speeds up the heart. Sympathetic speeds up the heart rate. Sympathetic dilates your blood vessels. Sympathetic constricts your blood vessels but dilates your pupils, right? Like if you're in danger. Parasympathetic is more calming. It slows down the heart, slows down the breathing, but it increases your digestion. Craniosacral outflow, cranial nerve 3, 7, 9, and 10. 3 is the oculomotor, 7 is the facial, 9 is the glossopharyngeal, 10 is the vagus. Vagus means vagrant because it wanders to like 80 to 90% of the human body. And then S2, 3, 4 keeps the feces off the floor. The neuron is electrically excitable, unlike the neuroglial cells, which are not. And you should be familiar with the key portions to this, such as the dendrites, the cell body, the axon hillock, the nucleus, the axon. This is the nucleus of the Schwann cell. This is an entire Schwann cell right here. And the gap or the spaces like here and here between each Schwann cell is called the node of Ranvier. So the purpose of the myelin, laying down the myelin, it's like an insulation. It speeds up the conduction of the nerve impulse to get to the end, and when it reaches the end or the terminal, we have these end bulbs which will then release a neurotransmitter to the postsynaptic side, right? Maybe it's going to be a muscle, and now that neurotransmitter is going to be acetylcholine. Okay. So the functional unit of the neural system is the neuron. It's electrically excitable. We showed you the nucleus. We have, just like other cells, the rough endoplasmic reticulum. We have ribosomes for protein synthesis. There's neurofilaments, there's microtubules, there's dendrites, and axons. The purpose of the dendrites is that it brings in the data to the cell body, and then the axon sends the information away. Everything around that neuron in the middle are neuroglial cells. They're all supporting cells. So again, you can see the dendrites, it means tree. It's going to conduct the impulses towards the cell body. The dendrites are typically short, they're highly, highly branched, and they're unmyelinated. Okay? What is really neat about this picture is you can see the neurofibril here, and then you can see the Schwann cell, right? You can see the myelin sheath of that. It's kind of like wrapping around and around and around and around and around and around, and it keeps wrapping around, creating this insulation. When you lose that insulation, when you lose the myelin and it demyelinates, or there's an autoimmune condition of that, that's referred to as MS or multiple sclerosis. The axons conduct impulses away from the cell body. The impulses actually arise from an area called the trigger zone, which is right near the axon hillock, where you see the initial segment right there. Okay, that's where the action potential is going to arise. This area here, this axon collateral, is just a side branch coming off of it. And the swollen tips at the end are called synaptic end bulbs, and these contain, these are little vessels in those end bulbs that are filled with neurotransmitter. Again, if there's a muscle over here at the end, then perhaps that neurotransmitter is going to be acetylcholine, which is always excitatory at the neuromuscular junction. In terms of axonal transport, the cell body is the location for protein synthesis. It needs protein synthesis to repair neurons or to produce neurotransmitters. Now there's slow or fast axonal flow. One of them, which is the slow axonal flow, only moves the substances in one direction, and that's always away from the cell body. And the movement is about 1 to 5 millimeters per day, pretty slow. The fast axonal flow moves organelles and materials in both directions to or from the cell body, and it can move substances 200 to 400 millimeters per day. That's pretty quick. But this becomes a problem when it comes to the fast axonal transport, is how pathogens and microbes can enter the neural system, such as Clostridium tetani. So if someone steps on a nail and it happens to be rusty and it happens to have this Clostridium tetani, and that spreads and moves quickly into the brain and into neurons, it will disrupt the motor neurons, creating tetanus, which is very painful muscle spasms and lockjaw. So bacteria that enter the body through a laceration or a puncture wound, it doesn't mean that the person's going to get tetanus. It's just if their immune system is compromised and weak and it can't handle it, then there is a problem. It becomes more serious when the lesion is closer to the brain, right? So if the lesion is like in the arm or in the chest or in the neck or in the fingers, it's going to move to the brain quicker than if it was stepping on something, from the foot. From a functional classification, neurons can be classified both structurally and functionally. So from a functional standpoint, it could be a sensory neuron, a motor neuron, or an interneuron. So a sensory neuron is afferent. Afferent and sensory means the same. It's going to transport the sensory data from the skin and from the muscles, the joints, the organs to the CNS. After it processes it, when it sends the information out, that's efferent information. Interneurons are the middleman. It's the middleman between the sensory and the motor neuron. So if I were to draw, see if I could do this for you here. Let's say here's the spinal cord. And that's a cross section of it. Let's say this is dorsal, and that's the dorsal root ganglia. You'll know what that means shortly. And then from here out to, let's say, a skeletal muscle. Let's say this is striated, and this is skeletal muscle. So this is motor, this is sensory. Sensory comes in, motor comes out. Motor is also efferent, whereas sensory is afferent. Now, the interneuron, or the association neuron, is relay. It's going from here to here. That's the association, or the interneuron. It's going to take the sensory info, relay it to the motor side. Okay, 90% of the neurons are association neurons or interneurons. So that's the functional classification. Structurally speaking, I wanted to touch upon this as well. In terms of sensory receptors, we have touch receptors here. We have deeper, and these are more superficial. We have Pacinian corpuscles, which are lamellated. These are deeper for deep pressure. Nociceptors, like noxious. These are pain receptors. And then in the skin, we have these cutaneous mechanoreceptors, cutaneous skin, superficial, and these tactile epithelial cells. So we have different types of sensory receptors. From a structural perspective, they can be classified as either multipolar, bipolar, or unipolar. Multipolar is the most common, right here. It's the most common, where you have several dendrites and you have one axon. Bipolar is where you have one dendrite coming off the cell body and you have one axon off the cell body. These are found in like the special senses, like the eye, the ear, sense of smell. And then the unipolar is, it starts off as a bipolar and then the cell body kind of pinches off. When it pinches off, here's the cell body. Okay, this is always sensory, and the classical example of that is the dorsal root ganglion. Remember that picture I drew before? Let me do it again, where I drew the spinal cord, and then I made the letter H, or the butterfly, in the middle here, and then I had a sensory coming in, a motor coming out, and then I drew this little lollipop stick there, and I said that's the DRG, or the dorsal root ganglia. Oh, that was horrible, dorsal root ganglia. So this is posterior, and this is anterior. So this is the anterior root, which is motor, and this is the dorsal root, which is sensory. And this is the dorsal root ganglia. So the dorsal root ganglia is sensory, because afferent information comes in this way, which is sensory. Okay. Association neurons, or interneurons, they can be named for the histologist, like Purkinje cells, or they can be named by their shape, like this one, which is a pyramidal cell.
The primary function of the nervous system is to provide rapid communication within the body to maintain homeostasis. This function underlies behaviors, thinking, and control of organ functions. The nervous system uses neurons to provide its functions, which include sensory input, association or decision-making, and motor output. Sensory neurons detect external and internal stimuli, and then convey sensory information into the spinal cord and brain. Association neurons make decisions based on sensory information provided via the sensory neuron. Motor neurons carry information from the association neurons to effector cells and organs. The effectors implement the decision of the association neurons.
Good. That was a very good introduction there. Okay, so neuroglial cells that I showed you a little bit earlier in this presentation, they make up half the volume of the central neural system. They're much, much smaller than neurons, but they're 50 times more numerous. These cells can divide, and there are 6 different ones. There's 4 in the CNS, and there's 2 in the peripheral neural system. The 2 that are located in the CNS, the 4 that are in the CNS are astrocytes, oligodendrocytes, microglial cells, and ependymal cells, and the 2 in the PNS are the Schwann and the satellite cells. So here you can take a look and you can actually see, let's see if we can highlight a few of these. Here's the oligodendrocyte, and here's a microglial cell. Here's an astrocyte. Here's another microglial cell, an ependymal cell. And then what are some of the other words we learned? Here's a neuron. Here's an oligodendrocyte, which myelinates in the CNS. Here's an axon. There's the myelin sheath, the gap between the Schwann cells, or the nodes of Ranvier. There's another astrocyte. Here's the piamata, cells of the piamata. Here's another neuron. Here's a capillary, and what helps make up the blood-brain barrier is an astrocyte. Here's another astrocyte. Here's a phagocytic cell. Here's a cell that makes CSF. Really nice picture from this view, just showing the axon right here, and you can see kind of like a jelly ring, the Schwann cell just keeps wrapping around, and that creates the insulation, the protection, if you would. Okay, so the Schwann cell is what myelinates in the PNS. The astrocyte is going to form the blood-brain barrier. It's involved in metabolizing neurotransmitters and regulating potassium balance. It's involved in action potentials maintenance. The oligodendrocyte is going to form the CNS. It's analogous to the Schwann cell, but this one's going to myelinate in the CNS, whereas the Schwann cell myelinates in the PNS. And here's the Schwann cell. Okay. The microglial has phagocytic role. It's going to clear away dead cells. The ependymal cells are responsible for producing CSF, and they're going to line the ventricles, which are cavities in the cerebrum. Satellite cells, these are primarily supporting neurons in the PNS. There's another great picture of the Schwann cell. You could see the axon in the center. Here's the nucleus of it, and it wraps around, it engulfs it like a macrophage kind of, and it just wraps around, wraps, creating this insulation. This just shows the process of engulfing them, kind of like a Pac-Man, and it keeps circling and circling and circling until you have that full insulation there. Oligodendrocytes myelinate axons in the CNS. Broad, flat cell process are going to wrap around the CNS axons, but the cell bodies do not surround the axons. There's no neurolemma that is formed in the CNS. In the PNS there is, but not in the CNS, which means that little regrowth is can take place after an injury. It's really not possible due to the lack of a distinct tube or neurolemma. Okay, one of the other things that you can see here on the left-hand side, this is a cross-section of the spinal cord, and again this would be dorsal, and there would be a dorsal root ganglia here, and then motor information comes out to the front to, let's say, muscle, organ, or gland. So this is sensory, this is motor, this is dorsal, and this is ventral. Okay, so these horns in the gray matter, you could see in the spinal cord, the gray matter is that butterfly in the middle. These are called dorsal horns, these are called ventral horns. And the white matter surrounds the gray matter. This and the white matter, they're called columns. So these are dorsal columns, these are ventral columns, and then these are lateral columns to the outside. All right? In the spinal cord, the gray matter is in the middle. That's the letter H, or the butterfly. White matter surrounds it. The white matter has tracts, which is a group of axons within the CNS. If you look at the brain, there's a complete flip-flop where we have white matter on the inside, right? All this, where I'm making the letter X, this is all white matter. And then the gray matter is all here on the outside. It makes up the cortex. Cortex means bark, like the bark of a tree. That's all gray matter there, okay? White matter is myelinated and gray matter is not myelinated, okay? All right, so that was a little introduction to the neural system.